Welcome back in today's video. In this video, we will learn about the agar, which is basically an excellent hardening agent of the media. Depending upon the physical nature of the media, it can be subdivided into three types. One is solid media, one is semi-solid media, and another one is liquid media. So there are three types of media based on the physical nature of a media. So in case of liquid media, there is only, it can be said as a broth. In microbiology, we should mention this term for liquid media. Liquid media means it's a broth. If nutrient media is present in a liquid form, then it is known as nutrient broth. Okay, so there is no hardening agent is present in this media. But in bacteriology, or if you want to isolate some bacteria in a bacterial plate, like this is a Petri plate, a glass Petri plate or plastic Petri plate. So we have to pour the media. So if it is a broth, means the liquid media, then it will not solidify in this plate, and the media that will be wasted. We cannot use this. To culture the bacteria, to get the pure, well isolated colonies of bacteria, we need a solid medium to grow the bacteria. So that a solid medium means there are some important thing or a hardening agent which is present in the liquid media. We have to add it with the liquid media to make it in a solid version, so that we can pour it in a glass Petri plate. It will solidify at some temperature, and then we can streak or spread the bacterial solution, and then we get isolated colonies of bacteria, which is very much important for the isolation of any kind of a bacteria from the natural sources. So the solidifying agent, what is the solidifying agent here? In microbiology, agar is most important solidifying agent or hardening agent that is used in the liquid media to make the media as a solid one. So now agar, that is basically extracted from red algae. Okay, agar is originally extracted from the red algae. Now, what is the nature of this agar? Agar is basically a polymer. Okay, it is basically nature-wise a sulfated polymer. Okay, it is first found in red algae and extracted from that. And what is the chemical nature? Chemical nature is it is a sulfated polymer. It contains three types of sugars. Majority is the galactose. What kind of sugars are present in this agar? One is D-galactose. Number 2 is 3,6-anhydro L-galactose, and the third one is D-glucuronic acid. Okay, so these three sugars are present. This is basically a glucuronic acid. These two are the major sugar constituents which make the polymer of this, and this is basically a sulfated polymer. So this is a chemical nature of the agar. Now, another important thing is why it is advantageous to use the agar as a solidifying agent in the microbiology. There are various properties which are very much important, and it actually helps us to use the agar as a solidifying agent. What are the properties? Number 1, the agar basically melts at about 90 degrees centigrade. At 70 degrees centigrade or 80 degrees centigrade, 60 degrees centigrade, the agar is not easily melted. That is why the melting point. It basically at about 90, 85 to 90 degrees centigrade, basically 90 degrees centigrade, when the temperature reaches, the agar will melt. Now the second one is, as it is not easy to melt the agar, it is also true that once the agar is melted, it only solidify when it reaches a temperature about 45 degrees centigrade. So at 60 degrees centigrade or 70 degrees centigrade or 50 degrees centigrade, the agar is not solidified. At 40 to 45 degrees centigrade, normally the 45 degrees centigrade, usually it is experimentally proved, the agar that will solidify. And at 90 degrees centigrade, which is just the double of this temperature, this solidifying temperature, the agar will melts. So it is not so easy to melt the agar and not so easy to solidify the agar. At a specific temperature, it will solidify, and at a specific temperature, it will melt. So the melting or the solidifying temperature, as it is already specified, so it's very advantageous for us to use the agar as a solidifying agent for the bacterial culture media. And another important thing, which is very, very important, the bacteria, the microbes, they cannot degrade the agar present in their growth media, because a nutrient agar means the nutrient broth plus we have added the 2% agar. The bacteria which grown on this nutrient agar media, it cannot degrade the agar. It is also experimentally proved. So that is why there are 3 important properties of this agar which make them as a useful hardening agent for the microbial growth media. Hope you like this video. Thank you so much for watching this video.